Hello, and welcome to the fourth lecture in our Adaptive Immunity lecture series. And here we'll begin talking about the major histocompatibility complexes and antigen-presenting cells. We talked about these in the beginning when we talked about nucleated cells and macrophages. Okay, we talked about them quite a bit actually, and so now it's time we talked about them. And apologies. And again, remember that our red blood cells are not nucleated cells and they would not have these major histocompatibility complexes, but most of our other cells are nucleated, and that's why we kind of make that distinction here. All nucleated body cells. Okay, so it's hard to kind of think of any others that aren't nucleated other than the red blood cells. So that's kind of what they're alluding to there. Okay, so the major histocompatibility complex 1 is found on all cells other than those, all nucleated body cells, and 2 is found on macrophages, dendritic cells, and B cells. Okay, they also have MHC1s, right, because they are nucleated body cells. Okay, but their MHC1s performs a function, and the fact that they have an MHC2 performs a different function. Okay, and so here you can see that all nucleated body cells have this type. Okay, class 1 and class 2. Okay, and this tiny distinction is how we have a distinction here, but they kind of do the same thing in that they recognize antigens. Okay, so we also have antigen presenting cells, and the dendritic cell will phagocytize a bacterial cell and bring it into what is known as a phagosome. Okay, in the phagosome, the lysosome fuses to this, and they release all of their enzymes, and they start to degrade the bacterial cell. This is known as the phagolysosome. Okay, if we can just piece it together from phagosome and lysosome, phagolysosome. Okay, so we've eaten it, and now we're trying to lyse it. Okay, inside of a body here. Okay, and that is shown here, engulfed, and then here the lysozymes fuse and begin destruction. During the destruction, antigens are produced. Hey, we recognize this as an antigen. This reacts with a component of our immune system, and let's put that on our MHC2. Okay, so we're going to send it out, put it onto our MHC2, and we will present that. Okay, so that is an antigen presenting cell, which are our dendrites and macrophages. Okay, and T cells recognize these presented antigens. Okay, remember we talked about cytotoxic T cells and helper T cells. They are recognized by the T cells and perform their function. Okay, so T cells and cellular immunity. Okay, so this is how we shorthanded nucleated cells. Okay, so nucleated cells is cellular immunity. Okay, we're going to use T lymphocytes. Okay, T lymphocytes are a type of white blood cell. They are made in the red bone marrow, and they mature in the thymus. Okay, the thymus is an organ that rests upon the heart. Okay, and it is surrounded by a bunch of tissue, makes it kind of hard to see. But this is where the T cells in the thymus are matured. And we already kind of showed you helper T cells and cytotoxic T cells, okay, based on our examples from our nucleated cells and our macrophages. Okay, note we haven't talked about fluids or humoral immunity yet either. Those are our B cells that we saw. Okay, so helper T cells and cytotoxic T cells are associated with MHC2 and MHC1 as we see. Okay, and so since we're in our cytotoxic section, we will learn more about these ones. Okay, and then we'll learn about the helper T cells when we learn about the macrophages. So the T cell receptor is very similar to an antibody, and it kind of performs the same function. Again, it has a variable region that is the antigen binding site, and it's very specific to the specific antigen. Okay, and this recognizes both MHC1s and MHC2s. Okay, and if we're going to activate the T helper T cells, we need to have presented the antigen, right? So we already need to know that process. This antigen presenting cell has engulfed the pathogen, destroyed it, found some antigens, and now are presenting it. The naive helper T cell sees, binds to that antigen with its barcode reader, the CD4. Okay, it reads the CD4. The CD4 reads the antigen that is presented, and from then it undergoes differentiation and proliferation. Okay, and they make a couple of T cells, and then some even form into memory helper T cells, which remember and stay alive a lot longer. Okay. We also have the cytotoxic T cell version, and how we can activate those is very similar. Remember, this is for nucleated cells. Okay, so here, instead of a dendritic cell presenting the antigen, the infected cell presents the antigen. It's like, hey, I'm sick, my proteins and functions are all out of whack. Here's an antigen. The cytotoxic T cell picks that up, reads it with its CD8 barcode scanner. The CD8 reads that this cell is infected, and they release perforins and granzymes in the matrix and destroys it. Perforins, think of it as perforating it. If we're going to perforate the cell membrane, we will open up holes in it and destroy it. Okay, and so that is an activated cytotoxic T cell. Okay. All right, and those are how our T cells work, and we will get into the B cells and humoral immunity and the rest of our adaptive immunity. Notes will be in the next video.